When our grandparents were having babies, the only way to know the sex of the baby was to wait until the baby was born. But then came advances in medical technology, like an ultrasound which at 14 weeks revealed the sex of the baby. In the 70s, a procedure known as in vitro fertilization was invented to help couples struggling with fertility to conceive. Then in the early 90s, thanks to great advances in scientific knowledge, it's now possible for you to know the gender of your baby way in advance of birth. As part of the in vitro fertilization process, preimplantation genetic diagnosis, also called PGD, allows you to determine the gender of your baby before the embryo is implanted in a woman's uterus. Having the ability to predict what a baby is before birth is great. But what actually determines if a baby is a boy or a girl, and how is it different in humans versus other animals? Well, most mammals, as well as a variety of insects, have XX or XY pair of chromosomes, which makes the chance of producing a male or a female 50-50. One chromosome is handed down from the mother, and one chromosome is passed down from the father. A set of X chromosomes produces a girl, and an X and Y chromosome together produces a boy. Because women only possess two X chromosomes, they can only give the egg an X chromosome, but men have one X chromosome and one Y chromosome in their sperm cells, thus they can give the baby an X or Y chromosome, which determines the sex of the baby. Birds also have sex chromosomes, but they act in completely the opposite way as humans. Male birds have two copies of a large, gene-rich chromosome called Z, and females have a single Z and W chromosome. The tiny W chromosome is all that is left of an original Z, which degenerated over time. When cells in the bird ovary experience meiosis, which is a special kind of division that produces eggs with just one set of chromosomes, each egg cell receives either a Z or a W. Fertilization with a sperm, all of which bear a
Z, produces ZZ male or ZW female chicks. Birds can control the sex of their chicks. We would expect that during meiosis, random separation of Z and W should result in half the chicks being male and half female, but female birds are different than their mammal counterparts. Somehow the female can control whether the Z or W chromosome gets into an egg, and most bird species produce more males than females on average. In humans, it's the sperm that determines whether an embryo is a male or female. In birds, it's the mother who decides the sex of the chick. But in reptiles, it's a totally different story. Sex-determining mechanisms in reptiles are broadly divided into two main categories: genotypic sex determination, GSD, and temperature-dependent sex determination, TSD. Species in the genotypic group, like birds, have sex chromosomes, which in reptiles come in two major types. Many species, such as various species of turtles and lizards, like the green iguana, have X and Y sex chromosomes, just like humans, with females being homogametic, that is, having two identical X chromosomes. Males, on the other hand, are heterogametic, with one X chromosome and one Y chromosome. Other reptiles that follow the GSD have a system, identical to one found in birds, with Z and W sex chromosomes. In this case, which governs all snake species, males are the homogametic sex ZZ and females are the heterogametic sex ZW. In temperature-dependent sex determination, instead, it is the environmental temperature during a critical period of embryonic development that determines if an egg becomes a male or female. This thermosensitive phase occurs after the egg has been laid, so sex determination in these reptiles is at the mercy of the ambient conditions affecting egg clutches in nests. For example, in many turtle species, eggs from cooler nests hatch as all males, and eggs from warmer nests hatch as all females. In crocodilian species, the most well-studied of which is the American alligator from Mississippi, studied both in the laboratory and in the field, scientists have concluded that sex is determined by the temperature of the egg during the second and third weeks of incubation. Eggs incubated at 30 degrees Celsius or below during this time period produce female alligators, whereas those eggs incubated at 34 degrees Celsius or above produce males. At 32 degrees Celsius, 87% of the hatchlings were female. Moreover, nests constructed on levees close to 34 degrees give rise to males, whereas nests built in wet marshes close to 30 degrees produce females. These findings are obviously important to wildlife managers and farmers who wish to breed this species, but they also raise questions of environmental policy. The shade of buildings or the heat of thermal effluents can have dramatic effects on the sex ratios of reptiles in both low and high temperatures. A common view is that temperature-dependent and genotypic sex determination are mutually exclusive, incompatible mechanisms. In other words, a reptile's sex is never under the influence of both sex chromosomes and environmental temperature. This model indicates that there is no genetic predisposition for the embryo of a temperature-sensitive reptile to develop as either male or female, so the early embryo does not have a sex until it enters the thermosensitive period of its development. This paradigm, though, has been recently challenged, with new evidence now emerging that there may indeed be both sex chromosomes and temperature involved in the sex determination of some reptile species. Apparently, in animals where both occur, certain incubation temperatures can reverse the genotypic sex of an embryo. For example, there is an Australian skink lizard that is genotypically governed by X and Y sex chromosomes. A low incubation temperature during the development of this lizard's egg reverses some genotypic females' XX into phenotypic males so that they have only functioning male reproductive organs. Therefore, in this species, there are both XX and XY males, but females are always XX. A slightly different example of this temperature-induced sex reversal is found in an Australian dryden lizard, which has the ZW system of sex chromosomes. In this species, high incubation temperature during egg development reverses genotypic males ZZ into phenotypic females. So females can be ZZ or ZW, but males are always ZZ. Reptiles in which both incubation temperature and sex chromosomes interact to determine sex may represent transitional evolutionary states between two endpoints, complete GSD and complete TSD. It is quite possible that there are other species of reptiles with more complicated scenarios of temperature reversal of chromosomal sex. There are certainly many known examples of fish and amphibians with GSD, in which both high and low incubation temperatures can cause sex reversal. In these cases, all genotypes from ZZ and ZW to XX and XY are susceptible to reversal by extremes of incubation temperature. Bonalia viridis, the green spoonworm, is a marine worm noted for displaying exceptional sexual dimorphism. That the sex is dependent on where the Bonalia larva settled. The female Bonalia worm is a marine, rock-dwelling animal with a body about 10 centimeters long. She has a proboscis that can extend over a meter in length. The male Bonalia, however, is only 1 to 3 millimeters long and resides within the uterus of the female, fertilizing her eggs. If a Bonalia larva settles on the seafloor, it becomes a female. However, should a larva land on a female's nose or enters the female's mouth, it then migrates into her uterus, and there it differentiates into a male. Thus, if a larva lands on the seafloor, it becomes female. If it settles on a proboscis, it becomes male. Another example in which sex determination is affected by the location of the organism is the case of the slipper snail Crepidula fornicata. In this species, individuals pile up on top of one another to form a mound. Young individuals are always male. This phase is followed by the degeneration of the male reproductive system and a period of lability. The next phase can be either male or female, depending on the animal's position in the mound. If the snail is attached to a female, it will become male. If such a snail is removed from its attachment, it will become female. Similarly, the presence of large numbers of males will cause some of the males to become females. However, once an individual becomes female, it will not revert to being male. For some, it may be difficult to decide which species is the best at gender determination, but if I had to pick one, in my humble opinion, it would be the bird, because the mother bird gets the option to choose if she wants a boy or a girl, and since she is the one carrying the baby, I think that's extremely fair. What do you think? Should the mother get to choose, or should it be random like mammals, or dependent upon the current temperature of the environment, like the reptiles.
Like, comment, subscribe.